Hello, Chem 212 students, and welcome back to our second and final lecture. There will only be two this week for mass spectrometry. So, in this chapter, chapter 14, we're talking about infrared spectroscopy, and that was the first lecture. And now we're going to do the second lecture, which involves mass spectrometry. So, again, the value of infrared spectroscopy was it allows us to determine what types of functional groups we have in the molecule and which types of atoms are bonded and how they're bonded, whether it's single, double, or triple bonds. Mass spectrometry is going to be a complementary technique that helps us get information that IR can't. Namely, it will tell us more about what what chunks of the molecule are connected to what other chunks of the molecule. So mass spectrometry is, in a sense, a destructive technique. A lot of times what it does is it will smash the molecule apart, and we'll get to see various pieces of it. And by determining what the various pieces are, we could see how they might have fit together in the molecule. So we can get an idea of the complete construction of the molecule, not just what individual types of bonds are there, but how the molecule was put together, which atoms were bonded to what other atoms altogether. So it will tell us the molar mass and the formula for the compound. So we'll get a peak that tells us the total mass, so we can see the mass, and it will give us an idea of what atoms in total are in the molecule. So in the mass spectrometer, as I said, it's a kind of destructive technique. Compounds vaporize, it's then ionized, and electrons knocked off. And often when it's ionized, it will undergo fragmentation. And based on the way that the molecule fragments, we can figure out how the pieces were originally put together. So it's kind of like smashing something inside a box, and then opening the box and seeing how all the pieces fit together to find the final thing that you smashed inside the box. So the masses of all the ions will be detected, and we'll get an idea of the masses of the entire molecule, and also all the various pieces that came about due to the molecule being broken apart in the ionization. So the most common method of ionizing molecules is by so-called electron impact, and so the sample is bombarded with a beam of high-energy electrons. Those slam into the molecule and cause another electron to be knocked off, and so what you'll get is a so-called radical cation. So it's what a radical means is that you have an uneven number of electrons, and this occurs because one of the electrons was knocked off. Also, you have a cation because the entire structure is now missing one electron, and so now it's missing, and it has basically one more proton altogether than it has electrons. So altogether, it has a positive charge, and so this is referred to as a radical cation. Uneven number of electrons because it lost one, and a positive charge. So the mass of the radical cation is going to be the same as the parent compound. And so if we detect the radical cation in the mass spectrum, we can determine what the mass of our initial molecule that we are analyzing is. So the radical cation doesn't always remain intact. Sometimes it completely breaks apart because of the electron impact. But if we can see it in the spectrum, we will label it as the molecular ion, and it will be labeled as M plus dot. So the plus dot indicating the radical cation, and M indicating our molecule of interest. So when this radical cation is formed, it will immediately fragment because radicals are very, very unstable, and they're going to immediately react. So it will separate, and one of the molecules, or one of the pieces of the molecule, will get the electron, and there will therefore be neutral, but we'll have an uneven number of electrons, and this will be referred to as the radical part here. And then the part that didn't get the spare electron, well, that one will just now have an even number of electrons, but it will be a cation. Okay. Now, for the cation, the part that's the cation, that's the part that will be detected by the spectrometer. The neutral part will not. The way that the mass spectrometer works is that it has a magnetic field, which will and a way of accelerating this. So it'll have a negative plate somewhere, which will attract this positive carbocation, and then that will be deflected by a magnetic field. And based on the arc that it takes and the deflection, the mass to charge ratio will be able to be determined. Usually the charge is 1, so the mass to charge ratio, it gives you directly the mass. But we do indicate, instead of mass, we say on horizontal axis of mass structure, we're going to see m over z, which is the mass versus a charge. Usually the charge is 1, so the mass is just the mass. Neutrally charged fragments cannot be detected. But sometimes, when you have a situation like this, the electron can go either way. So for one molecule, this side becomes the cation, the right side. For a neighboring molecule, the left side becomes the cation. So often, as long as there's not any preference for making a carbocation on one side or the other, often we'll see both. However, if one produces a more stable carbocation, we'll see a lot more of that one as a result of the fragmentation. So the mass spectrum is going to show us a relative abundance of each cation that's detected. So that's important. Cation, okay? So again, if one potential cation is more stable than the other, we're going to see a much larger peak associated with that cation. And that might just indicate that that's a more stable cation, or it could indicate that there's more of that piece. It could indicate either one. Often, the base peak, the molecular ion, will be the tallest peak, and so we would label it like here. This is probably the molecular ion, so we would label it m dot plus. It's also called the parent ion a lot of the times. And so the molecular ion here is at 16, which makes sense, right? Because the mass of this CH4 is 12 for carbon, if it's a carbon-12 isotope, and 1 for each hydrogen, assuming they're all hydrogen 1. And so 12 plus 1 is 16. Now you can see we got a little peak here over here, right, at 17. What's that all about? Also, we have other peaks that are smaller than that, so let's talk about those in a moment. We will. But here we're focusing on this molecular ion, which is 16 for the methane. Now you notice that we did see a lot of peaks with m over z that were less than the molecular ion, and these were produced when the molecular ion was ionized and upon its subsequent fragmentation. So here, if we smack a methane with electron and do electron impact, we lose one of the electrons in the bond, and at that point, this molecular ion will fragment. So this is the molecular ion that had m over z is 16, but then it fragments, and here we have a hydrogen atom, radical, it only has one electron, and then we get the methyl carbocation. And so this is the one that weighs 15 here. And so that's why we see a fragment that weighs 15. And then if there's even more fragmentation, we could get losing another hydrogen where we get m over z is 14, and 12, and 13, and 12, and so on. The lowest we get is 12. The 12 would be the bare carbon atom as a cation. So mass spec is a sensitive analytical method. It's used very, very broadly in a great number of applications. For example, in pharmaceutical industry for drug discovery and reaction monitoring. So this again can tell us what types of pieces we have here, and allow us to characterize a drug that we are attempting to synthesize and make sure that it was synthesized correctly, and also to monitor dynamics biologically. In biotech, we use it for amino acid sequencing, determining the structure of large proteins is a very common technique here in biotech. They use a special type of sampling, which they call electrospray ionization, where basically it's attached to little liquids. But you don't need to know the details of that. But if you're going to be studying proteins later, you may use electrospray ionization. Mass spectrometry is a pretty common analytical technique to determine what is a sequence of amino acids in a protein. And you can see there's a lot of other situations in which mass spectrometry will be used. It's a very useful technique for identifying molecules in many different job environments or science environments. Next, we have a mass spectrum for the structure of benzene here. So one thing you're going to notice about benzene, well, if you look at the last one, for the methyl, there was heavy fragmentation of the methyl. Although the majority of the molecular ion did not fragment before sampling, a lot of it did. It fragmented, it lost lots of hydrogens, as many as all four. When it comes to benzene, you'll notice that the fragmentation is much, much less significant. The molecular ion is right here, and it's much, much larger than any of the other peaks. And this just goes to show you the resonance stability of this the strength of the bonds in the benzene ring. It's very, very hard to break these bonds. It does not fragment significantly. So if we lose one hydrogen here, that can happen a little bit. But all of these bonds are very, very strong. Benzene does not tend to break. So when you have an aromatic ring here, the take-home message is that you don't get a whole lot of fragmentation when you have the aromatic ring. There's a little bit, but not much. Whereas if you have a straight train hydrocarbon like this, so this was benzene C6H6. This is hexane, so C6H14. All single bonds, and you can see those single bonds break quite easily. And so we've got all kinds of fragmentation. Often when you have a long hydrocarbon chain, you don't even see the molecular ion. Like the molecular ion, if we counted it here, we should see it around 6 times 12 plus 14. 6 times 12 for carbon plus 14. That would be 72 plus 14. So that would be 86. So we expect to see a peak around 86. Of course, it's, let's see, 1, 2, 3, 4, 5. What's 1, 2, 3, 4, 5? Whoops, sorry, C5H12. Okay. I miscounted the carbons here. So 5 times 6 plus 1 times 14, or 14 times 1. And so we get 5 times, 12 for carbon. So 5 times 12 is 60. 12 the mass of carbon, 1 for the mass of hydrogen, so 14, for a total of 74 here. So we would expect to see a rather large peak at 74, which would be right here, but we see no peak at all. And so you'll see that in some molecules, like the benzene, with these very strong bonds that are difficult to break, we have very little fragmentation. Almost all the molecular ion peak is by far the largest peak. In this case, though, it fragments quite easily, all single bonds, and there's almost no molecular ion peak. So the molecular ion peak would be right here if it survived, but very little of it survived. And so we don't really see the peak here. So when we want to analyze the mass spec, the first thing we want to do is find the molecular ion. So we want to know what the whole molecule is, so we can then analyze how it was broken up and how it fragmented. So we look for the M over Z of the parent ion and the molar mass of the compound and see if a peak is there. Now, the big thing you want to look at with the molecular ion peak is if you want to see if it has an odd mass or an even mass. If it has an odd mass, that's a dead giveaway for a nitrogen atom or a phosphorus, but usually a nitrogen. So if we have an odd number of nitrogen atoms, we are going to get an odd number for a molecular ion peak. So that's a dead giveaway. So first check, is there an odd number for the molecular ion? If we have an even number, that doesn't mean we don't have any hydrogens. It means we have an even or any nitrogens. It means we either have no nitrogens or we have an even number. But the odd mass is what you're going to look for. That will be a giveaway for an odd number of nitrogen, usually 1. Next, you want to look at the peak that's right next to the molecular ion. So this is called the M+1. So the M+1 peak, you'll often get an M+1 peak in hydrocarbons because about 1% of all carbon atoms weigh 13 instead of 12. So for almost all of these methanes here, the mass is going to be 16. 1 times 12 plus 4 times 1, 16. But if the carbon weighs 13, then you're going to have 1 times 13 plus 4 times 1. You're going to have a mass of 17. And so you're going to see a little peak at the M+1. So this is the M+1 because about 1% of these carbon atoms weigh 13 instead of 12, and so the total mass is 17. And so you can see the size of this peak is about 1% of the size of the molecular ion right here, and that is because about 1% of those carbon atoms weigh one more. Okay. Now, the more carbon atoms you have in your spectrum, the larger this M+1 peak will be relative to the molecular ion, because all you need is for one of those many carbon atoms to have a mass of 13 to give you a peak here at the M+1. So here we can see in decane. Decane has 10 carbon atoms in it. So decane is C10H2 times 10 plus 2. So C10H22. It has 10 hydrogen atoms. So if one of those, 10 carbon atoms. If one of those carbon atoms weighs 13, you get a peak here. Since there are 10 of them, there's about a 10% chance of having one carbon atom that weighs 13. And so you can see that the M+1 peak for decane is about 10% the height. For every carbon atom, you essentially add 1% to this M+1 peak. And so if you have 10 carbon atoms, then it will be 10%, or it will be rather a 9-to-1 ratio. So it's actually slightly higher than 10%. It will be 10% out of 100. So here, 90% of them have the mass of 142, but 10% of the parent molecule has a mass of M+1 or 143, because there are 10 carbon atoms. 1% for each carbon atom. Another thing to look for are the M+2 peaks. So M+2 peaks are a dead giveaway for a halogen atom, especially depending on their ratio. So chlorine comes in two common isomers, chlorine that weighs 35 versus chlorine that weighs 37. And so for this chlorobenzene here, the parent ion is going to have a mass of 112. So here's the parent ion, and then we have an M+2 peak right here. And so the fact that this M+2 peak is about a third of the height of the parent ion peak, the molecular ion, that tells us that we have a chlorine atom, because of the chlorine atoms, about a quarter of them are going to be 37, and about 3/4 of them are going to be 35. So you'll get about a 3-to-1 ratio of the peaks for the molecular ion versus the M+2 if you have chlorine. If you have bromine, so for either chlorine or bromine, you're going to have an M+2 peak. But if that atom is bromine, your M+2 peak is going to be almost the same height as the molecular ion, because bromine has two common isotopes. One weighs 79 and one weighs 2 more, it weighs 81. And they're almost the same abundance. And so you'll get an M+2 peak that's just slightly smaller than the molecular ion peak when you have bromine. So far, we've checked the molecular ion peak, and then we checked the molecular ion +1, checking for how many carbon atoms there are, and we can check the molecular ion +2 to see if there are any halogen atoms in the molecule. So now we're done checking the molecular ion. Now we're going to look at the fragmentation pattern. There's a lot of things that can happen in fragmentation. So first of all, let's say we have the pentane again, and so here we have the molecular ion. So it's C5H 2 times 5 plus 2, 12. And we've knocked off one electron, and so we have the molecular ion. At that point, it's going to fragment, and it can fragment in lots of different places. An interesting thing about this, I'll just share with you guys, is when I got my PhD, about half of my research thesis was about trying to figure out the relative amounts of the molecular ions, how they fragment either at the first bond or the second bond. And then I did this for a great variety of these straight chain hydrocarbon molecules. You might be surprised to find that the relative fragmentation had not been directly observed, and so I had a special mass spectrometry technique that allowed us to cool down the molecule immediately after it reacted figure out where it would break with exposure to heat. Anyways, these are not breaking with exposure to heat; they're breaking due to electron impact, ionization. But when the molecular ion breaks, it can produce a methyl radical, or it can produce this ethyl or this rather methyl cation, ethyl cation, propyl cation, or butyl cation, depending on where it fragments. And so this is going to be something you see very commonly. You're going to see an M-15 peak if the counter ion that's not detected is methyl. You're going to be wanting to be looking at the 15 mass units below the molecular ion. This will tell you had the fragmentation where a methyl group came off that wasn't detected. You detected this fragment, so the methyl fragment came off and was not detected. If you have a peak at 29 unit mass overcharge units less than the molecular ion, that's a loss of an ethyl radical. M-43 is the loss of a propyl radical, and M-57 is the loss of an ethyl radical. So after you've looked at the molecular ion, you're going to want to look at these mass regions to see if you've lost any of these hydrocarbon fragments. So here we are again with pentane, and we can see again for pentane the molecular ion would be right here. It's at 72. So the molecular ion is 72 here. And you can see that it's very small because we've had a lot of fragmentation, right? And so we want to look for what's 15 less than 72. So we say 72 minus 15. Okay, so that's going to be 62 minus 5. So that's going to be 57. And so we look for the 57 peak. This is 55. 55 right here. 55. 56, 57. So 57 is right here. This is the M-15. We can see we get a pretty significant fragment here. Then we would do 72 minus 29 here. So this is M. We got M minus 29. So that's going to be minus 20 here will be 52. Minus 9 will be 43. And so this is 40 peak right here. So this is the 40 peak, 41, 42, 43. So the M minus 29 is right here. And we get a very significant fragment there. And then we look for M minus 43, which is 29. There's a significant peak here. And you'll notice something. That itself has had a elimination of 2 hydrogen atoms. And so that's something to think about as well. We'll be looking at that a little bit. So there's a significant amount of the elimination of 2 hydrogen atoms. Why is that? We'll think about that too. And then here we have the M-57 peak for that. That's for the loss of the butyl. And so after you've checked the molecular ion, you'll want to check for those situations there. So look at this. So this molecule here. This molecule is not a straight chain. So in this case, there's lots of spots that it could fragment, right? It could fragment. We could fragment here. We could fragment here. We could fragment here. But there's going to be a preference to fragment where I'm about to put this blue line here. So there's going to be a preference to fragment right here. And the reason why is because when it does fragment, it produces the radical cation. And if the electron goes over to this primary radical, the result will be you'll have a tertiary carbocation. And we know about the stability, the relative stability of tertiary carbocations. So we expect to see a rather large peak here because it will be quite stable here to produce this tertiary carbocation, and it will be produced in a much greater fashion here. There will be much more of this tertiary carbocation versus these other less stable carbocations produced. And so this would give us an M-43 peak due to the loss of the propyl radical. The propyl radical here would be C3, H7. So that's going to be 3 times 12 plus 7 times 1. So that is 36 plus 7. That is going to be 43. And so we would want to look very carefully here for the M-43 peak and expect that it's going to be large. Now, another thing that can happen is when you have the radical cation, this is going to fragment in some fashion, and sometimes there can be a rearrangement that will allow for a more resonantly stabilized cation to be formed. This will be especially true when you have an alcohol. So there's going to be special things we can look for when we have an alcohol that will help us to know that we have an alcohol, and this is due to particular way in which the alcohol will fragment when you get the radical cation. So if we have a radical cation here, we've removed one electron from the oxygen, so we can see right here that electron would have been right here, and it's not there anymore. And so what's going to happen now is there's going to be a kind of rearrangement that occurs that's going to allow for some resonantly stabilized result. So what happens is that we look at the alpha carbon, there's going to be a cleavage between the alpha carbon and a beta carbon. We call this the alpha cleavage. And the result is that we're also going to get a double bond to the oxygen. And this is favorable because we've got the cation on the oxygen and we've got a pi bond that's adjacent to the cation. So we have a resonance structure that can occur here. These pi electrons, we move them onto the oxygen, and we have a carbocation here. This will be especially prevalent if it produces a tertiary carbocation on the carbon, right? And so this resonance stabilization can be quite strong. In addition, the other type of fragmentation that can occur is that you can have an elimination of water, and both of these will happen to some extent. So we have the radical cation here, and then we have what essentially amounts to an E2, and we get an elimination of the hydrogen and the oxygen, and in the end we get this double bond here, right? So we can think of it kind of like, remember the way we thought of these before in chapter 8 here. Something kind of grabs the hydrogen, then this leaves, and then this goes, right? And of course it would have to be protonated first. Realistically, it actually protonates itself, but in the end, what matters is the hydrogen and the OH are lost. So you lose water, and you end up with a double bond. So the first thing you want to do when you suspect that you have an alcohol in your structure is to look for an M-18 peak. This, especially if it's a tetra-substituted double bond like this, it's going to produce quite a stable molecule. So you should get a pretty significant M-18 peak, 18 here being the mass of water, right? And so if you spot that, you should also look for the evidence of a nice and strong peak that results in the alpha cleavage here of the alcohol. And that will be your confirmation of the alcohol. You should see both of these things happen. Amines can also undergo this alpha cleavage here. It happens in the same way. Effectively, what happens is you end up with a double bond between the nitrogen and the alpha carbon. So here we had double bond between the oxygen and the alpha carbon. Here we have double bond between the nitrogen and the alpha carbon. And then you get a breaking off here right between that alpha carbon and a neighboring carbon. And again, this will give you a pretty significant peak due to the resonance stabilization. If you have a carbonyl, you're going to get something very fancy. So what a carbonyl is when you have a carbon double bonded to an oxygen. That's called a carbonyl. So here we have a radical cation that's a carbonyl. So you can see that there's one electron missing right here, right? And so that electron has been removed, so it's a cation. And these, remember, in real life, these molecules are rotating around and spinning around. And if you have a carbon that is 3 carbons down, so here we're calling the alpha carbon, in this case, is attached to the carbon that's on the carbonyl. That's the terminology for carbonyls. The one that's called alpha is actually the one next to the carbonyl. So you can think of this carbonyl as this whole group. Alpha means next to that group. The same way, this is amine, it's its whole group. The carbon is the carbon next to that group. So here the carbon counts as a part of the group. So we call the alpha carbon next to that. Then the beta carbon is here, the next one over, then the gamma carbon. A hydrogen on the gamma carbon can actually pop on over and go on to the carbonyl carbon. And this is favorable because this gives you an oxygen here that has 3 bonds and 1 lone pair, so it's positively formal charged. Also, you have here a you have if this bond is rearranging here, what you've got is that 1 extra electron is here on the alpha carbon. So look what happens. So these are fishhook arrows. All of these are fishhook arrows. They indicate the movement of 1 electron. That's something I should have mentioned earlier. These are indicating the movement of 1 electron rather than 2. And so here we can see this. We got 2 electrons that go into this double bond, right? Here we've got 2 electrons that go into this double bond. And there's 1 electron on the alpha carbon. And so that allows for some resonance stabilization here. We have a carbocation adjacent to a pi bond, and we also have a radical here, a radical electron that can be used to create the pi bond. And so we have this here. This is creating a pi bond right here. And then we have the positive formal charge still on the oxygen here, but it's single bonded. And this is referred to as a McLafferty rearrangement. Notice that we get 2 pieces. Let's analyze this one more time. So the hydrogen goes over here. We end up with the bond to the hydrogen here. This ends up here as a double bond. And so we get this very stable doubly bonded structure out of that. And then the bond is breaking right here. And so here's where we get that stabilized, resonantly stabilized cation. That's the other fragment. And so we're going to see this fragment here. And so we're going to see a significant loss of some even numbered fragment. That is the fragment that would be broken off if you broke the bond between the alpha and the beta carbon here. This can be a little bit more difficult to spot here. So you have to watch out for that. But a big tell will be that you're going to have a big peak where you have a loss of an even mass group. This is going to be even mass. So this is kind of like the M-18 for the alcohol right here. You're losing an even mass, but you're going to lose a larger even mass. And so that's something to look out for. Especially you'll look out for it if your IR spectrum that goes along with the mass spec indicates that you have a carbonyl. You should be looking for this McLafferty rearrangement. So the common fragments we should be looking for, these are the alkyl fragments here. So the methyl radical, the ethyl radical, the propyl radical, and so forth. The butyl radical. So these are all alkyl. Loss of alkyl radicals. They're odd numbers. Notice they're all odd. Okay. Whereas if you have water, you should look for the M-18 peak. Or if you have an alcohol, that loss of water will tell you there's an alcohol. And if you lose a large even number, then that's indicating a McLafferty rearrangement, which will occur when you have a carbonyl, a ketone, or an aldehyde. So for the mass spec, the most obvious advantage of it is it tells us the molecular ion. So it determines the molecular mass, so we can often figure out the molecular formula. So you may recall that I had previously told you that saturated hydrocarbons have a formula that is Cn plus 2. For example, if we've got propane, this is C3H8. The N is 3, and 2 times 3 plus 2 is 8. Now, if you have a double bond, though, that's going to be different. So notice here, if I have propene, CH2, double bond, CH, CH3, this is propene. This is going to be C3H6. Every time we have a double bond, we're going to have 2 less hydrogens. So a double bond is 2 less hydrogens. So saturated is this. This is saturated. If you have a double bond, it's 2 less hydrogens. If you have a triple bond, it's 2 less hydrogens. And for every ring that you have, every ring is going to give you 2 less hydrogens. Sorry, and the triple bond is not 2 less hydrogens. It's 4 less hydrogens, meaning 2 again is what I meant. So every time you have a double bond, you'll have 2 less hydrogens than saturated. For every triple bond you have, you'll have 4 less hydrogens, and for every ring, you'll have 2 less hydrogens. So if we have, for example, a ring and a double bond, so this has 6 carbons, a saturated with 6 carbons would be, let me look at my next slide here. Yeah, I'll show it on the next slide as well. Saturated with 6 carbons would be C6, H2 times 6 plus 2, H14. But if we look at this structure here and fill in all the hydrogens, we'll see that we have C6, H1, 2, 3, 4, 5, 6, 7, 8, 9, 10, H10. So we had a double bond which lost us 2 hydrogens and a ring which lost us 2 more. So that's why there's 4 less hydrogens than the saturated compound. So that what I just described was referred to as the degree of unsaturation. Every pi bond or ring counts as a degree of unsaturation. And so that's minus 2 hydrogens. The number of hydrogens is reduced by 2. So again, here with the double bond, we have with saturated pentane is C5H12. With a double bond, it's C5H10. With a triple bond, it's C5H8. These are so the ones with the double and triple bond are referred to as unsaturated. Here, these are various isomers of C4H6. So if it were saturated C4, we would have C4H2 times 4 plus 2, 10. 2 times 4 is 8, plus 2 is 10. But we've got 2 degrees of unsaturation on these, so they have only 6. So this can be achieved by 2 double bonds. That will give us 2 degrees of unsaturation. 1 triple bond is 2 degrees of unsaturation. If you have 2 rings, each one is 1 degree of unsaturation, so that would be 2. And if you have a ring and a double bond, that's 2 degrees of unsaturation as well. And there's a term that your book uses, 1 degree of unsaturation, is called 1 unit in the hydrogen deficiency index. I tend to use this terminology much more, degree of unsaturation, but your book also refers to it as the hydrogen deficiency index, or HDI. When it comes to atoms that are not carbon and hydrogen, this is how they affect the HDI scale. So if you have a halogen atom, it replaces a hydrogen atom. So for example, here we have C2H5Cl. We can say the chlorine atom is replacing a hydrogen atom, so this is equivalent to C2H6, and 2 times 2 plus 2 is 6, so this is saturated, right? So it's the same as having C2H6. So this is saturated. So you just, when you're trying to see if it's saturated or not, take the halogen atom, pretend like it's a hydrogen, and if it's saturated, then it's 0. And for every 2 hydrogens missing, you have 1 degree of unsaturation. Oxygen will have no effect on the hydrogen deficiency index. So if you have an oxygen, you can essentially ignore it. If you have the right number of hydrogens while ignoring the oxygen, you're saturated. For every 2 hydrogens you're missing, you have 1 degree of unsaturation. For nitrogen, for every nitrogen you have, you're adding 1 extra hydrogen. So notice that if there were no nitrogen atom here, if there were just a hydrogen, we'd have C. Here we have C2H6 saturated. But here, this is C2H7N. So what I do in my mind is I just cancel a nitrogen and a hydrogen. So I say, okay, for every nitrogen, I subtract 1 hydrogen. So I say this is equivalent to a saturated C2H6. So this is saturated. So in general, for a halogen atom, you add 1 hydrogen, which would be where it is. For oxygen, you ignore it. For nitrogen, you remove 1 hydrogen. That's the extra hydrogen that's attached to nitrogen, for comparison to a saturated hydrocarbon. Or you can use this formula, which I do provide to you. It's 1/2 times the hydrogen deficiency index, so the degrees of unsaturation will be 1/2 times 2 times the number of carbons plus 2 plus nitrogen minus plus the number of nitrogens minus the number of hydrogens minus the number of halogen atoms. If it's 0, then it's completely saturated. If it's 1, then you have 1 degree of unsaturation, and so forth. I don't use this formula though. I do it this way. I just replace the halogen atom with the hydrogen, I ignore the oxygen, and I remove 1 hydrogen for every nitrogen, and then compare to the saturated hydrocarbon. That's the way I do it. Yeah. So when it comes to analyzing mass spectra, so when we go into the lab, I'll show you guys a little bit more about analyzing mass spectra here. But that'll do it for this week's lectures, and then we'll talk about analyzing mass spectra in the lab. I hope you have a great week. See you later.